Hi. So, in previous session we have learnt about the concept of PRR. We have learnt various families of PRR like TLR, RLR, NLR. We have learnt in greater detail about the toll-like receptor. We have learnt about their ligand, and we have learnt about the signaling pathways activated by TLRs. If you remember, there are two major signaling pathway. One is that MyD88 dependent pathway and another is TRIF dependent pathway. So, after this signaling pathway, it is very important to look for the TLR and diseases, because after the discovery of this pattern recognition receptor, which is about approximately 3 decades back, happened in 1989 or 1996, 97, 98, it was quite well established. Thereafter, this field was expanded like anything, okay, and this field still it is expanding, and this field is quite promising in terms of understanding disease, okay. So, therefore, in this session I would like to discuss about this TLR and diseases, otherwise this is kind of incomplete. And most likely in another session I will also discuss about the TLR and therapeutics, okay. So, let us begin with this TLR and disease. So, here you can see it's a TLR4 mediated signaling, and if you remember, for TLR4 signaling the ligand is lipopolysaccharide, right, and this lipopolysaccharide is basically a very potent immunostimulator. If you remember, I have discussed in previous several sessions, okay. And this LPS sensing is not so simple, it is little complicated. You need LPS binding protein in which is present in soluble form in body fluid, particularly in serum or plasma, okay. And there is a CD14, and this CD14 is also a soluble factor, okay. And there is a one molecule MD2. So, this complex basically is sensed by the TLR4, okay. And then this TLR4 induces the shock, if you remember my previous session, okay. So, this shock is quite complicated condition, okay. So, if there is some changes in these gene, so that may result to the that is not necessarily result to the development of disease, it may be a protective, okay, for some situation. So, all those things I am going to discuss in this session. So, let us look at there is a polymorphism in TLR4, where this aspartic acid, which is at 299 position, changed to the glycine, okay. So, if this kind of polymorphism is there, then the individual has a decreased response to the inhaled LPS, and there is a decreased incidence of atherosclerosis, okay. So, atherosclerosis, you might be aware that this is a generally we talk in context with cardiovascular diseases, right, and it is simply the thickening of wall of these arteries. So, there is a arteriosclerosis in which any artery of the body can involve, and this thickening is basically is mainly caused by depositation of various things. It could be a cholesterol, it could be a lipid, and whatever makes the wall thickening, that narrows the lumen of this artery, okay, and then this will this may reduce the blood flow, and if it is happening in some in major artery in the heart, then that result to the cardiovascular disease, okay. So, if this mutation is there, then there is a this is a kind of a good news that there will be a decreased incidence of atherosclerosis, okay. Increased gram negative bacterial infection. So, you can understand if there is a less response from this TLR mediated signaling, then that will increase the possibility of multiplication of gram negative bacteria, right. It is a quite compatible or quite obvious, okay. So, if this mutation is there, then there will be a possibility that the individual is kind of susceptible to the gram negative bacterial infection, okay. There is a increased severity of atrophy. Atrophy is basically the literal meaning is the thinning, okay. Atrophy can be associated with various things, atrophy in neurons, atrophy in muscles, wasting of muscles and all those things, okay. So, there is a possibility that there will be increased severity of atrophy, decreased airway hyper reactivity. So, this is also possible. So, all these conditions are associated with TLR4 mutation, this D299G, okay. There is a also some polymorphism is associated, that is CD14, and this mutation is basically present in promoter region, okay, where the C is replaced by T, okay. And if this mutation is there, then there will be a increased response to the LPS. Probably this mutation may increasing the expression of CD14. I do not know precisely, but most likely if you see the response, because this is a increased response to the LPS, means there is a more CD14 protein available in extracellular fluid, okay. So, that will of course obviously cause more response to the LPS. Another is this IREC4. If you remember, there is a this MyD88 recruits IREX. In previous session I have discussed, and then TRAF6, and that makes a kind of signalosome, a complex, and that eventually activate the IKK complex. So, this IREC, there is a IREC4, IREC1, and if you remember, I discussed about the IREC-M, which is a kind of negative regulator, okay. So, this IREC4 has a stop codon. If there is this polymorphism will be there, then that will introduce a stop codon. This is 287, 293, okay. And there will be a increased susceptibility to the gram positive infection, okay. Another is this NEMO. So, NEMO is a one of component of IKK complex. So, IKK complex is consist of IKK alpha, IKK beta and IKK gamma, and IKK gamma is also known as NEMO, okay. So, if there is some polymorphism in this NEMO, then there is a possibility that the individual is more susceptible to the viral and bacterial infection, okay. Another is I kappa B alpha, and there is a mutation here. You can see that the amino acid serine is converted into the isoleucine at 32 position, and if this mutation is there, then there is a possibility that that will impair the T cell memory, okay. So, these are some polymorphism which is associated with TLR signaling. So I will talk more some more TLR and their polymorphism and their association with the disease, as you can see in this table. So in TLR2 there is a one polymorphism, which is basically conversion of threonine to the cysteine amino acid. So this is at position 597, and when this mutation will be there, this is a kind of beneficial to the host in terms of susceptibility to the leprosy. Okay, so these individuals are protective from the Mycobacterium leprae infection somehow. Okay, and on another hand, they maybe have a more chances to have a tuberculosis. Okay. There is a one more polymorphism associated with TLR2, that is this is R amino acid change, which is arginine, which is converted into the to the Q amino acid, and this basically these individuals are kind of susceptible, or there is a predisposition of staphylococcal infection. So this is associated with TLR2. TLR4 I have already discussed, and I will talk about one more this polymorphism, which is basically the threonine is converted into the isoleucine at 399 position, and these individuals may have some as I told you, there will be a reduced risk of arthrosclerosis. There is an increased risk of systemic inflammatory syndrome and septic shock. Okay. However, it is not associated with rheumatoid arthritis. Okay. So these are some mutation which is playing very important role in protection or development of disease. Okay. There is a TLR5, a dominant negative stop codon mutation that will basically increase the susceptibility to the pneumonia caused by Legionella pneumonia, and negatively correlated with Crohn's disease. Crohn's disease, if you remember, this is a inflammatory bowel disease. Okay. So there are please remember, any disease or most of disease, I will say, particularly the complex diseases such as cardiovascular disease or some metabolic disease, cancer, generally all these diseases are multifactorial. It is caused by multiple factors. So you cannot pinpoint that, okay, only this factor is responsible. In general, these diseases are happened by, there are so many factors. Okay. So here I have just discussed the role of TLR in susceptibility or protection in disease. So I will move forward, and then I will talk about there are few more molecules of TLR signaling, that is MAL. If you remember, MAL is also known as TRAP. Okay. And this is a kind of linker between the TLR, particularly in case of TLR 1, 2 and 6 and TLR 4. Okay. So this is a kind of linker. This MAL or TRAP is linker between TIR domain of these TLR with the MyD88. Okay. And there is also some mutation are reported, which is associated with some disease. One is that you can see that there is a serine amino acid which is replaced to the leucine at 180 position, and this mutation shows the protective effect against developing tuberculosis and severe sepsis, malaria or SLE. Okay. However, it is not associated with rheumatoid arthritis, and heterozygote may have a lower risk of developing chronic Chagas cardiopathy. Okay. So this chronic gas cardiopathy is it's a parasite induced cardiopathy, and this parasite is Trypanosoma cruzi. Okay, so this is a chronic gas. It's a chronic condition. Okay, it takes a long time, basically, that disturbs the cardio function. Okay. Another molecule is IRF5, and IRF5 plays a very important role in induction of uh, uh, type 1 interferon as well as inflammatory cytokines. Okay, and there are some mutations or some polymorphism is reported, and it has been shown that these polymorphism is basically lead to the increased susceptibility to the SLE. Okay. And the last polymorphism, if you see the in case of IRF5, this leads to the increased protection against SLE. Okay, so these are quite complex. It's understanding that the deeper biology is not so easy. So we because this polymorphism is present in human, and we cannot make a genetic manipulation in human. Okay. Of course, we can do it in mice, and we can dissect out the basic mechanism, but it's quite challenging. Okay. Another molecule is IRAK4. There are several SNPs including C877T, where the cysteine is converted into the threonine, and this is associated with recurrent Streptococcus pneumoniae and Streptococcus aureus infection. Okay. There is a decreased inflammatory response to the LPS, and there is an increased IgE concentration in serum of patient with CRS and asthma. So what is CRS? It's a chronic rhinosinusitis. So this chronic rhinosinusitis is a there is a rhinovirus infection, or which is also cause rhinosinusitis. This chronic word means if this situation is persisting for more than 12 weeks, then we call it as a chronic rhinosinusitis. And this is that there is a more release of mucus from the nose, stuffy nose. Okay, and all those things, and it is not getting cleared, so then we call it as a chronic condition. Okay, and this is also associated with the IRAK4. Okay, the MyD88, which is a very key adapter molecule, okay, in TLR signaling, there are some minor variant in 3' UTR, and these individual has a decreased antibody response to the measles. Okay, so this measles is again a viral disease, which is you might be knowing that it is caused by single stranded RNA enveloped virus. Okay, and there is a spots all over the body. Okay, particularly in trunk, in hand, and so on. And this is associated with quite high fever, about 105 Fahrenheit. Okay, so in this session I will stop here, and I have discussed there are some genetics are playing very important role in susceptibility or protection against particular disease in case of this TLRs. Okay. In next session I will talk about the TLR and therapeutic, which is you will see that how this discovery of TLR, and now we have some molecules which we are using for treatment of various diseases, and those molecules are basically activating or damping the TLR signaling. Okay. So thank you very much.